everybody. So it is as usual for spring. I'm saying now as usual, a beautiful day. I driving in today, just looking out that the trees have become greener, and there was this red cardinal sitting. As I was driving, I could see this spot red cardinal in the beautiful trees. And it's like, yes, spring is here, spring is alive, things are, everything is flourishing around us. So it's a beautiful day in Northwest Arkansas. So those of you who are on Zoom or maybe new here coming to this seminar series today, my name is Rodrigues. I am Associate Vice Chancellor and I'm the Executive Director of the Institute for Integrative and Innovative Research. In case you don't know it, we call it IQDAR. We are driven by purpose, and our mission is to pioneer solutions to wicked problems that don't have easy answers, that require you to take risks, that need you to converge multiple sectors together and come in from academia, industry, government, non-profit areas, all put together to come up with solutions to these big wicked problems. And as we have talked about it earlier, we want to be the leaders, the recognized leaders, the preeminent leaders in this space, so that we can bring about transformative change, not just within a university, but within an ecosystem, within the region, within the state, within the nation, and really by making, giving answers to big, difficult solutions to the world. And at the end of it, our purpose is to be able to have a societal impact and improve the health and well-being of everybody. I've also often told you all that there are 5 words that remember when you think about IQDAR, and of course integrative, which is right there in the name of our institute, convergence, economic development, at scale, and societal impact. So if you can remember those, then you know who we are. As we have done through over the series, we have brought in experts from many different aspects of trying to look at how we can bring together food, health, cyber security, all of that put together in order to come up with a mission of how do we solve big wicked problems. And one of those big wicked problems is integrative health. And today I'm very pleased to have a wonderful speaker with us who has already spent yesterday telling us about how to navigate spaces to put grant proposal in. I think she spent dinner time with 5 eager assistant professors, peppering her with questions. And so the person I'm talking to you about is Professor Deborah Rodrigues. She's the Ezekiel Cullen Professor of Environmental Engineering at the University of Houston. And she's going to be talking to us about a very interesting topic. Before I tell you about the rest of her credentials, let me say that how intriguing your topic is. She's going to look at how plant-based polymeric nano-drug delivery systems, usually we're thinking in terms of human nano-drug delivery systems, but this is plant-based nano-drug delivery systems can be a potential game changer for gut infection. So perfect example of how we are talking about bringing food, health, high-end technology all pulled together. This is integrative health. It is bringing all things together. So here's a solution for medicine, precise, gradually being targeted. And so it's also precision medicine. It is precision nutrition. It is, I'm just waiting to hear all about it. But this comes because she has herself such a broad background to be able to converge all these different things in this one talk itself. She has a bachelor's degree in biology and biology education and a master's in science in microbiology. She came from Sao Paulo, Brazil, and a PhD in microbiology and molecular genetics from Michigan State University. So you can see science coming in there, right? And biology coming in there, and molecular biology and genetics coming in there. And then she did a postdoc at Yale. And then later, she starts to put together all of these different things with some from bacteria, initially looking at bacteria stuff, and then all the way to carbon nanotubes in soil and transportation and filtration of it. And for the past many years, she has been in civil engineering. So you see the science and the engineering all being put together. And then for 2, 3 years, almost 3 years at NSF, looking as a program director across in the TIP directorate, which we know about TIP directorate because we ourselves have a convergence accelerator here, Meredith Hopkins, our fearless PI leader on that. But pulling together not just basic science, not just engineering, but moving stuff so that it is actually deployable and making impact. That is what the TIP therapist is doing. So she has the inner understanding of that and extremely valuable experience of being a program director at NSF where you're looking at all these different. So it gives me really a huge pleasure to invite you, Professor Rodrigues. It's all yours.
Thank you so much. Wow, that was a very nice introduction. Thank you. I appreciate you. Really covered the breadth and the broad areas that I actually work on. Although I'm not going to talk about water or the environmental engineering part, I'm going to be talking about the fighting foodborne illnesses and everything started also as part of the environmental engineering problems is emerging antibiotic resistance. And just to give a little bit of background, antibiotics is increasingly being used across the world, and US is one of the main users, and it's growing as well. But China is going to lead in a few years that usage. Europe also is one of the main users, but they are a little bit more conscious and they use way less than we do. And most of the antibiotic use, whether we like or not, is with livestock. And humans as well is the next one. So there's a lot of antibiotic being used there. So you might be thinking, so what? We need antibiotics. It's good. I mean, we are sick. We need to treat it, right? Well, the problem with using a lot of antibiotics is that all that is put in, unfortunately, the antibiotic doesn't get degraded necessarily. Some of them they get partially, but it ended up in feces or manure that is applied in the land, and that ends up contaminating soil and groundwater. And with all that, you ended up creating microorganisms that are antibiotic resistant as well. And that ultimately will affect humans and the animals as well. So one thing that we want to think about it, so how we can actually circumvent the antibiotic resistance? That's a very hard question to answer and to solve. And one way is really simply don't use antibiotics or reduce the use of antibiotics, right? But economically not viable because we actually need antibiotics to really have a healthy livestock. Another aspect that we have been using in my research group is preparing nanoparticles for drug delivery. And why nanoparticles for drug delivery could be a good thing? First of all, you can have those particles that are more specific to a site contamination. So if you have an infection in one organ, you want to target it. You want to actually have the infection being treated there. So it's not spreading all over your body. And because we are made of microbes, literally, we have microbes everywhere in the gut, in the skin, you name it. And we don't want to take antibiotics that affect the whole microbiome. You want to really go to the point where you have the infection and get the antibiotic really tackling just that problem and not affecting the whole microbial community that we have in our body. That also protects us from other types of contamination, infections in general. And another thing that you want to make sure that you have the drug, but you also have a continuous delivery. So you don't have to keep taking antibiotics over and over and over. And then the more you take, the more you release in the environment, and the more you have this spread of antibiotic resistance that can be happening. So the idea is really, at the end, with that mechanisms, is actually to reduce the amount of antibiotic that we are using and the amount of antibiotic that is being released, to also reduce the antibiotic resistant emergence. So that's kind of what we had in mind as a hypothesis. If we can make that happen, we'll be able to not completely solve the problem of antibiotic resistance, but at least reduce the fact that we are already making in the environment and in antibiotic resistance per se. So this one, simply using less, is not practical. So we are going to be talking about that system. Now, my approach has been working with polymer nanoparticles for drug delivery because they typically, you can select the type of polymer and you have a variety of them. And you can have even polymers that are natural, that might be even safe and better for the organism as well. And the nice thing about polymers too, you can actually tune it. You can make modifications, you can add functional groups, and to make it better for site-specific delivery, or even to uptake better the drug. And you can also increase the amount of drug that you can load in those nanoparticles. So there's a lot of benefits of using polymers. And the approach that we use is comparing these three polymers. PLGA, which is polylactic glycolic acid, is the gold standard. So a lot of people already use this because they already know that it works. It's a good way of encapsulating in pharmaceutical products for encapsulation. They use PLGA. So it's the gold standard polymer that people use, but it's a synthetic one. We wanted to explore something that is more natural, that comes from agricultural aspects. So we decided to investigate lignin that comes from plants and zinc that comes typically from corn as well. And they are nice because they are typically biocompatible, or they are inert to the organism. They can have very low cost because they are really abundant in nature. It's a plant you grow, and you can use even their waste product. People that are not using the rest of the plant, you can actually collect and prepare and synthesize those nanoparticles. And they have all different functional groups that you can also modify and do whatever you want, basically. The sky is the limit. Let's put it this way. So we select these three polymers to compare. This is the gold standard and the two new ones to actually investigate. And in this case, in this study, we also use a specific antibiotic, erofloxacin, which is a fluoroquinolone antibiotic, commonly used for swine, cattle, poultry, so for prevention of intestinal diseases with E. coli, Salmonella, etc. But this talk, I'm going to be focused on this little guy here, the pigs. And most of the cells and things that I'm going to present is going to be about pigs. And the reason why we selected pigs is twofold. First of all, I don't know if you're aware, the genetic makeup of pigs is extremely similar to humans, like 99% of their genetic makeup. Of course, we don't look like them, but we are very similar genetically speaking. They also have a very similar digestive tract as us. And they can eat anything like us. They are omnivores. So there are a lot of similarities, if you will. And we had twofold interest, really, like for swine investigation, for livestock investigation, but also to understand whether this could be a model for humans as well. A lot of people use mice, but mice is so different in so many ways from humans. And we know that for drug delivery, they might not be the best model for it. So there are twofold ideas on this study, was really to understand whether pig could be a good model, and also for agricultural purposes as well. So just to give an idea about the overall talk, what I'm going to be talking about. So we synthesized all the nanoparticles, and we did the characterization to show that they were actually uptaking the antibiotics, and they were stable in the solution, and their size were actually a good size, a nano size that cells could uptake as well. do endocytosis in the sense that when we give to the pig, it will not actually be floating around in the gut, but it will actually enter the cell where the bacteria actually infects, because the bacteria also gets inside. The E. coli, it gets inside the cell and infects, and then start the process of being the infection process. So we wanted actually to see whether these antibiotics, these nanoparticles would get inside and start killing the bacteria inside the cells and not kill the microbiome that is in the gut. So we did that investigation to look the effect of the infection, and also we look on the effect as well of these nanoparticles in the gut microbiome. Because as many of you know, the gut microbiome is the one that actually helps regulate a lot of different diseases, colon cancer, diabetes, and etc. So we really don't want to mess up with our microbiome in our gut. That's the bottom line. And that's what we want to actually understand. So let me start with the synthesis and characterization of these nanoparticles. We use different approaches, but basically what we did, we got the polymer and the antibiotics and inorganic and aqueous phase, and the formation of the nanoparticle occurred under high pressure homogenization. And then we did evaporation and cryopreservation of the nanoparticles, so we could actually store for a while and do the use of as we needed, basically. And they were kind of similar for all of them. But if you want details about all the different syntheses, you can also check out those papers where we publish the detail of each one of them. But basically, that was kind of the preparation. And we were able to get nice nanoparticles for PLGA, lignin, and zinc. And just to give an idea about how they look like, so we got a kind of a size close to 100. Zinc was the smallest one. PLGA and lignin, they were kind of similar size, and the polydispersity index, which we were actually looking whether those particles would be dispersed in the media. So for better, you don't want them to aggregate and then clump all together, because then the drug delivery is not going to work as well. So we want the particles to be more dispersed. And so that level was actually showing that there was monodispersed particles. And the drug loading changed quite a bit. The best one that loaded the most was lignin, followed by PLGA. Zinc was a low loading drug, but still, you're going to see it was still efficient. But the nice thing about having them small, typically in this range of size, we know that nanoparticles might undergo endocytosis, and so we can get inside the cells, basically. And so, but we double check that by actually labeling the nanoparticles with Trixie. And we were able to see that over time, we actually got particles going inside the cells. It's not on top. When you do actually confocal and you do the layers, you actually see that they are really inside the cells. So the cells really uptake and get those particles inside. Now, the question is, are they actually getting inside and being toxic? Are they able to actually fight the microorganisms that are pathogenic? So that was our next question. We actually look up for that, but we also look at the drug release, how they were releasing the drug over time. So the first thing that we saw, because remember, we wanted not to keep giving antibiotic, but have over time treatment with the same dose that we would give. So we investigate also the drug release at the body temperature, and we saw a biphasic type of release for the nanoparticles. One of them is in the first hour, you have the loosely tracked drugs in the particles that were released, and then you have over 75 hours still release. So we didn't receive a sustainable release of drugs. We also looking at, this was in PBS, but we also compare with a more complex and simulated gastric system. There were some releases difference, but still was sustainable over time as well. And so meaning that the particles were able to do that. Then the next thing that we did was actually try to understand whether, okay, we prepared those particles, what is the concentration that would be effective when they are encapsulated on those nanoparticles? So we look at the different concentrations of the nanoparticle, the drug encapsulated in the nanoparticle, and how much we would have at least 50% inactivation of microbes on that. And so what we saw is that around this range between 0.12 and 0.2, we got inactivation about, and that line is 50%, inactivation of E. coli enteropathogenic microorganism. Now, for Zane, was actually interesting because when we had just the enrofloxacin, lower concentrations were not even enough to activate at 50%. But when we added to the Zane, that same concentration, we got almost 100% inactivation. So there was kind of a synergistic effect with the Zane and the antibiotic that reduced even the need of higher concentration of the antibiotic. And we tested with, I'm not showing here, but we tested with other gram-negative bacteria like Salmonella, and this effect was exactly the same. Although it doesn't work for gram-positive because you also test with Clostridium difficile. So it's really something that happens with the interaction of the gram-negative cell wall, just as a side information for you. But that was something that was quite interesting, that encapsulating actually allowed us to even reduce the amount of antibiotic that we needed to inactivate a pathogenic microorganism. That was very exciting. And overall, just to summarize these results, so the range of amount of antibiotic that we needed, whenever we had 3 antibiotics, about 0.14 microgram per ml. But then when you add the Zane, you could even use lower concentrations. And the Enro needed quite more, even though the loading, if you remember, was pretty high at the beginning. You were able to encapsulate quite a bit of antibiotic on it, but again, we still needed a much higher. So that was actually interesting to see that the nanoparticle really can play a role as well in the interaction with the antibiotic. But then we want to know also, can we use with the cells? Are the cells okay with those nanoparticles and all? So what we investigated, so we got the pig cell line. Again, we are working with pigs and trying to understand how they behave. So this pig cell line is actually primary cell. So they got a little piglet, and we got from the intestinal cells, we scrape off the cells from there to grow and cultivate the cell line. Actually, we bought the cell line from DSMZ. We didn't actually cultivate, but that's how they prepare and they got the cell lines, basically. And so we use the same concentrations that we test for the antimicrobial aspect. And overall, they were pretty okay with that concentration for PLGA and lignin and even Enro. In the case of the Zane, we really had to add a lot to start seeing something, which is actually not even realistic. So Zane was really, the toxicity was really low. No matter how much you added, we had to go like 320 microgram per ml, which is a lot. Nobody would have used that anyway. But just to give as a contrast as well, that cells, they might have different sensitivities to the different particles as well. And we were actually very excited that Zane was showing that high effectiveness against the enteropathogenic E. coli and very low mammalian cell toxicity. So we decided to go a little bit further with the Zane for more investigation. But just for this first part conclusion, what I want to show you is that the drug was, the nanoparticles were able to be encapsulated on the different materials and the different particles, and they could go endocytosis, which is nice. So we could actually have a target delivery to the cells. And we also had a slow release of the drug, so we could have more sustainable treatment without having to keep adding antibiotics to the system. And more importantly, we also saw that the Zen nanoparticle was the one that had the most non-cytotoxic properties and the lower need for antibiotic as well. So we ended up going with trying to investigate the infection prevention using with Zen. And we wanted actually to simulate the gut system. Most of the studies that you see in the literature, they actually use the flask, the 2D system, like you just grow in flask your cells, your cell line, and then you add either your antibiotic or your nanoparticles, and you do the investigations like that, right? However, the gut environment is way more complex than that, right? You have many different types of cells to start with. So, you have what is called the goblet cells. Those cells, they actually produce mucus for the gut as a prevention for infection, but also for nutrient absorption. So there are many roles that the mucus producing the gut has in that system. But when you do it to the culture, you don't get this type of complexity. You typically have one cell line there. And how realistic is that when you want to actually investigate the effect in the gut, the real gut? So we were like, the 2D system is the common one, but the gut is way more complex than the 2D system. So our approach was actually to try to simulate the intestinal cells. So we created a 3D bioreactor, and this is actually 3D printed. The whole setup was designed and printed in 3D. And we create also a mode with the villi, which is a scaffold. And here is a picture simulating exactly the size of what you see in the villi on the intestine, and with PDMS. However, PDMS is not a good substrate for cells to grow. They do not really attach to that. So we had to actually do some coating of the scaffolds with polydopamine, and we were able to successfully coat it. And then we did some additional tests. But this bioreactor, it had an inlet where you could have the media flowing through, and you would have the ability for the cells to grow and attach to the surface. And here's what it looks like, the whole system. So you have a pump that really simulates the flow of the food going through the intestines. And and in a speed that you'll typically see in the gut as well. And we have the little scaffolds, and then we grew the cells, and then you have the crypts that are the lower part, and then the villi there. And then you had the cell that were grown on it. And here's just an image with life in there, the green live cells. They were grown for 3 weeks, and you got a nice growth of cells and villi on it. So, but again, one thing that we didn't know is, would the cells differentiate in this system with the flow and everything? So we did hypothesize that the fact that we had a similar structure of the villi of the gut, the flow, all the conditions, and they would actually start to make some differentiation on this system. There are some research that show that cells, when they are in close proximity, they have a lot of cell signaling interactions that can occur that you typically don't see in 2D cultures. And that can actually lead to different signaling pathways with cells that can lead to differentiation as well. So that was our hypothesis, that in our scaffold with UDMS and polydopamine in the flow and all, we would have cells, for example, the stem cells, they tend to be in the crypts of the villi, and then the goblet cells here, they would be more on the villi, producing all the mucus. And of course, you would also have epithelial cells that are typically there. And so what we did, we grew in those scaffolds for 3 weeks, the pig cells that we got. And then we did some imaging with different markers to look whether there were presence of these cells in there and whether there is mucus production as well. And we confirm by qPCR the gene expression of those cells as well. So the first thing that we tested was like, well, are they even producing mucin? So we got a marker for that, the WGA. And the importance of having the mucus, which is, like I mentioned earlier, it's very different than what you typically see into these cells. And they are very important, especially for digestion, absorption, and immune defense against pathogens in general. They can be an additional barrier for infection that you typically wouldn't see if you don't have that, because then they are free to get in the cells, the pathogens. So what we did, we got the 2D flasks, and also our 3D systems with the villi. So this part here is actually the villi. It's just that it was a top view, but and here would be the crypts as well. And you see some mucin, very little, spread all over, very disorganized manner in the flat. But you do see quite a bit, specifically in the villi, which is typically in the gut is where you see more mucus being produced, because the goblet, they tend to be in the villi, not in the crypts. So, and then we actually said, okay, let's look whether the goblet cells that are producing this mucus are there, and where are they located. So when we actually use the marker for identifying the goblet cells that secrete those mucus, we saw that in the flat, there were just maybe a few, but almost nothing there. Very, very, very little. But again, remember, we're working with primordial cells, so there could have been some, but really, really little. As opposed to the scaffold, that we really saw a bunch of cells there, and exactly where they were supposed to, and where we saw the WGA marker as well in the villi. And then we look at the marker for the stem cells that can be differentiating in all the different types of cells. And this marker for the stem cells are typically found when you do the analysis of the intestine, is really on the crypt, so at the base of the villi. And when you look at the flat, we didn't see any of the stem cells there, but we did see in the villi, in the crypt here, the villi is here. So at the bottom of the villi, we start seeing actually the marker of stem. So 2 things, we got cell modification and we got cell localization in the right place, like it is in the gut in general. And we went even further and we look at the gene expression of the scaffold, which is the red color, versus the flask. And you can clearly see that the gene expression, the log fold change on expression was much higher when it was in the scaffold for both the marker for the stem cells and for the mucin, the goblet cells. So that confirmed what we were seeing in the images in the genetic aspect. So it was very exciting because we actually created a system that is really relevant to the gut system that we can investigate in vitro instead of actually injecting a pig and doing all the analysis in the pig, which is way, way, way more expensive and hard to get. We could actually analyze in an in vitro system that is much more close to what you see in the actual pig cell environment. So the question is, okay, we were actually investigating the nanoparticle, whether they could prevent infection and all, and we got our nice system there. So the idea is actually to compare whether this nanoparticle, we knew that there was a pig, but whether it was better in eliminating the pathogen with the nanoparticle versus just putting the antibiotic there. and how much the 2D system and the 3D system would differ. Because again, one, they produce mucus, that is an additional barrier to infection, and they also have different types of cells too, compared to the flat system. So we actually, the first thing that we did was try to identify, because we're not working in vivo, we had to actually identify in our system what would be the best infection dose. We didn't want to actually, we wanted to put the pathogen, let them infect, and then treat. But we didn't want to put the pathogen in a high dose enough that by the time we go and treat a few hours later, everything is dead. So we wouldn't treat anything because everything is dead. So we wanted to try to find exactly what would be the best condition that we had infection, but we wouldn't have all the cells killed, basically. So we had to find, so we did several tests using different dilutions and concentrations of cells in the pig cells. And here, the green color is the live cell staining. So here at this dilution, you can see most of them are dead. And then here we start to see quite a bit dead still. And then this one, we had a good number. So we decided to actually go with this dilution to do our testing, because we knew that there were infection happening, but we still had a good number of cells that we could treat and quantify, and which was the idea. So we selected those cells, and again, we had the flat, which is the 2D system, versus the scaffold. And one thing that we also looked is whether Zen by itself would also have any effect on treatment versus the non-treated, and they were pretty much the same. And however, when we have the enro in the Zen versus just the enro by itself, we can see that we got much more reduction of cells with the enro in the flat. But when we look at the scaffold, we got much, much, much better than the enro on itself. So we attributed actually to probably some interaction also of the Zen with the mucus that helped also the ingress of the nanoparticle in the system and in the treatment of the pathogen in the cells better than the enro. Because the mucus, you see, even here compared to the flat, the mucus is also a barrier even for the antibiotic. That penetration and diffusion through the mucus to get the cell and inside the cell for treatment, that was actually a barrier. So the system showed that, okay, the mucus is good if you have a nanoparticle, but for the free antibiotic, not so much. It still treats, it's still better than nothing, but there is that. Now, another aspect that people have concerns about nanoparticles are related to immune response. We saw that it can treat the microorganisms in the cells, it's great, but would that have also any effect on the immune response of the pig? Because you don't want to treat and cause another problem, right? You want to treat and solve the problem and not have any immune response on that. So we wanted also to see whether there was an expression, a different expression on the immune response of the cell, and whether you could see also a difference between the 2D system and the 3D system. And we hypothesized that it would be different because, again, you have different cells and you have mucus in one system and not in the other one. So it's not really the same thing, basically. So the markers that we tested was the TNFR1, that is part of the pathway for apoptosis and inflammation and survival of the cell. And we also passed the ICAM1 that is related to the pathway of recruitment and transmission of immune cells. They are really linked to the immune response, and the selection of those immune response markers was based on other studies that people showed that this type of response happened, especially with metallic nanoparticles, they actually saw that it can get expressed, but it can trigger the expression of this immune response for those particles. And so we started with those. And so when we look at the TNF in the scaffold versus the flat, one thing that you see right away, especially with the TNF alpha, is that it's a more homogeneous response. So didn't matter whatever you added, the scaffold had almost a similar response in relation to the infection and the treatment with the nanoparticles or the enro. So there was not much of a difference, although with the flat system, the 2D, you have a more different response. But what we saw as well is that overall, for both of them, the response of the markers was lower in the scaffold versus the 2D. And one thing that I want to point it out is that either Enro, Zang Enro, or even Zang by itself, they did not change the immune response. So the cell didn't really care about the nanoparticle being there or not. They were okay with that, basically. That's the main point. So the main thing is that from this part is that we were able to develop this in vitro system. And the nice thing about this in vitro system is that I use for nanoparticles, but I'm also using for toxicity of other food products like hydrocarbons in the food. When you cook, you produce a lot of different polyaromatic hydrocarbons, and you can actually investigate even toxicity of that. So this model, you can actually apply for many different types of toxicological assays and different things, basically. That's the bottom line. So we developed this system that can be applied in many different things, basically. And on top of it, I showed just one reactor, but you can do also multiple reactors. You can actually grow many cells and different cell lines, everything at the same time. And I didn't show here, but we also had a lot of success in producing in vitro system also from mice, human cells, and pigs. I'm not presenting here the data, but because we just started and we did some testing. And fun enough, if you start with primary cells, you also get differentiation with all the 3 cell lines as well. So this system can be applied to whatever you want that has a gut system. So as long as you have the primary cells there. So that was something nice from this work. And another thing that we saw is that doesn't mean that because you have less drug encapsulated that your effectiveness might be bad. We saw actually quite the opposite with Zay. We had less drug load, but we still had a lot of treatment, a much better treatment than the free drug by itself. And the nice thing is that the plant polymers, especially Zane, we saw no inflammatory response, at least for those markers. We need to explore further markers. But the fact that we didn't see any inflammatory response is very promising, that maybe food-based nanoparticles might be the answer for drug treatment. Now, the last part that I want to present is related to the gut microbiome. And the idea is really like to see whether these nanoparticles also they impact the gut microbiome and how they impact, and if there is any antibiotic resistance emergence happening, how much it changes the emergence of antibiotic resistance in this community if you use the nanoparticle versus the free antibiotic. So one thing about the microbiome, again, I want to highlight that for antibiotics, people already show that they can alter quite a bit the gut microbiome. And a good example is when you take antibiotic, a lot of people have diarrhea. They lose stools and all, because you kill not only the pathogen, but you kill everything that is in your gut, kind of, right? So you lose everything. So you alter, you change your microbiota composition. And with that, you're probably affecting even the whole human body and the metabolic activity. And they can cause dysbiosis, which is an imbalance on the system that you have a healthy system with good microbes are gone, then you might start having other types of side effects if you don't recover your healthy community there. And on top of that, because you eliminate a lot of microorganisms, the ones that are resistant, they will stay there. And you know the way that microorganisms, they exchange resistance in most cases is through plasmids. So they can transfer from a good microbe to a bad one, and then you create antibiotic resistance and even release to the environment that can contaminate food, the water, and then people start to get that. So there is this aspect. So the way that we investigate the whole system, again, we got the little pigs. We went to a slaughterhouse where they do not use antibiotics. It's a free antibiotic facility. And we wanted to start with a clean community. So we got they would slaughter the pigs and then just remove the parts of the intestines, the slurry from the intestine. We wouldn't work with the actual feces because the feces is very different than what you actually see in the guts. So we really want to work with the community that really have the one that is going to be affected the most when you receive antibiotics. And so we cultivated in a bioreactor as well. And we have different treatments that we use. We have the nanoparticles. And in this case, we didn't work with Zane specifically in this work. We're still working with Zane, and I don't have the results yet, so I'm not going to share, but we did with lignin and PLJ. We also tested with the free antibiotic and loaded nanoparticles with enrofloxacin. And we did not work with DNA, but we worked with the RNA because we want to see the microorganisms that were actually active and really functional on the community and understand what is going on, how this community is changing, if there are antibiotic resistant genes that is developing. And then we did sequencing and bioinformatics on that. So one nice thing that we saw with this community initially, we analyzed the whole community and we identified a core community. And nice enough, this core community is very, very, very similar to humans too. So again, another thing that it's similar, humans and animals. So that gives us the idea that, pig also could be a model also for the gut microbiome too. So that was one thing that we saw, and even with some other mammalians as well that had a similar digestive system as humans. We are investigating a little bit further that a community and comparing, and I'm not going to actually present this time, but just to let you know that there is a lot of similarity of some of the main bacteria that you find in the different communities. Another thing that we saw is that when you look at the controls over time, the empty nanoparticles of lignin and PLJ, we saw that the nanoparticle per se didn't do any effect on the microbial community. They just didn't really care about the particle being there. So the core community was still there, same abundance and all. So that's great news because we don't want the nanoparticles to have another effect that would add up to the antibiotic because we know that the antibiotic can create an effect. But we did see that with the free drug, and was expected that the core microbial community went down, several of them, but the most significant were these ones. And with the lignin enro, we did see some altered microbial community changes. Some of them went down and some of them were actually enriched. We got a little bit concerned about fused bacteria because what happened is that this bacteria in particularly have been associated with colon cancer. So increase of this specific bacteria was a little bit concerned because of that. I mean, it could indicate that there is actually a potential dysbiosis that is creating an imbalance on the microbial community that could lead to some negative effects to the health. And one thing that we saw with PLGA, but it was minor, is that, PLGA is a particle that can actually be biodegraded, and it's already known. And when they degrade, they produce a lot of lactic acid, and you do have a lot of bacteria in the gut that can use that. So there was a little bit of enrichment of some species related with lactic acid. But when you have enro only, PLGA with enro, there was a little bit of a change, but the core ones were still pretty much all there. So there was a much better conservation of the core microbial community of the PLGA enro versus the lignin enro. And when we look a little bit more related to the diversity and using some diversity measurements, we saw that the control here and the PLGA, they were pretty much, it's the purple one and the black one, they were pretty much similar. They didn't change over time. With the antibiotic, there was a little bit of a reduction, but not significant, as opposed to the one 48 hours and 72 hours that had antibiotic for the nanoparticle with lignin and enro as well. So there was just a small decrement on diversity on the PLGA enro treatment, but much more pronounced with the lignin here. But the main question is, okay, the community is changing and all, but how is the overall metabolism of your gut community is changing? So we did some analysis called PyCrust, which is a software to actually compare the different genetic information of these communities to see how they would actually affect, or if there is redundancy on the system, basically on your community. And what we actually saw is that overall for the genetic information processing, there was some variation, but not something that was striking. You still had enough abundance to have not so much as an effect on this side. So there was a general homeostasis on that. However, when we talked about some more specific metabolism functions, like some secondary metabolites or even glycan, cysteine, and lipids, what we saw is that especially with PLGA, we saw some increase and changes in the short chain fatty acids metabolism and lactate metabolism. And with lignin, we have a little bit of cross-feeding relationship changings as well that was happening. And just to summarize a little bit, so we know that those are plant-based, so it can be digested by some of the microorganisms, and even if they are released in the environment, they will be degraded as well. So the PLGA, the degradation is typically the formation of lactic acid and glycolic acid, and those types of components, they can be further degraded to increase small chain fatty acid, and that this type of short chain fatty acid is not bad actually. It can reduce gut inflammation and can help with the lipid metabolism and also antioxidant production, and even reduce the reduction of infections. And in the case of lignin, you can have some aromatic compounds that can be formed. And some aromatic compounds, they can also be good, but they can also be bad, depending on the type of aromatic compounds that are formed. So basically, that's what we saw related to the metabolism of the gut microbial community with those nanoparticles and the antibiotic. Now, the question is, how is the antibiotic resistance responding to things? And so one thing that we look at, we know that most of the enro treatment is mainly for they are fluoroquinolones, so they affect quite a bit proteobacteria. That's why we use E. coli, enteropathogenic. And but what we saw is that there was the treatment with empty nanoparticles and the controls. They actually didn't affect much that antibiotic resistance. And here you have the proteobacteria, which is the brownish. So there was a little bit of changes and reduction with the enro specifically here, but for the controls, you didn't see much of that. And even for PLGA with enro, you didn't see either. But one thing that we saw, if you look here over time, so enro in 24 hours versus 72 hours, we saw a significant increase on antibiotic resistant genes appearing in the community, as opposed to this is lignin 24 and 72 hours, it went down over 72 hours, and with polylactic acid, it also went down. So the fact that they were actually encapsulated in the nanoparticle, somehow it reduced the spread of antibiotic resistant genes, more so than the free load of the antibiotic. So that was actually something that we got very excited, because if we can reduce the antibiotic resistant emergence, that's kind of a big deal. And we did some correlation analysis as well to look at the red lines means the association of the enro by itself. And you can see that there are quite a bit of red lines here. So the majority of the antibiotic resistance genes were really associated with the presence of free enrofloxacin and not so much with the other nanoparticles. So that actually emphasized the benefits of using nanoparticles for drug delivery instead of just the free antibiotic. And just a little bit about what type of genes we saw. We were expecting to have gyrase genes being affected because that's the main mechanism. And because people have always studied with certain types of organisms and isolated culture, but not with a mixed culture like that, the effects of antibiotic in the gut. And when we actually did the analysis, to our surprise, there were a lot of genes related to antibiotic resistance that were actually expressed, especially with enro in particularly, and that we were not actually expecting. And but again, you were working with a very complex community, and we don't even know those microorganisms in detail and their response. So we look at the inhibition of protein synthesis and efflux pump, and all of these types of even enzymatic reaction associated antibiotic resistant genes were actually expressed. So as a final conclusion of this study, we saw a system that is physiologically relevant that we developed for the 3D. And we also saw that plant-based polymers can be very promising for drug delivery. And the fact that those polymers encapsulation can also reduce antibiotic resistance is also something that would be a great thing. And I would like to thank the USDA, the Welch Foundation, and EPA that they fund parts of this project, and the team that really helped this make it happen. Dr. Louis, Dr. Christina Savliov, Dr. Astet from LSU, and the students that did the work actually. And thank you.